In this lesson, we're going to take a look at microbial nutrition. We're going to talk about exactly what microbes need in order to grow and survive, and then that will help us understand the needs when we do start to culture them. And that culture is ultimately going to be very important if we do want to study them and possibly come up with ways to utilize them to our benefit, or possibly find ways to treat pathogens. So as we begin to talk about microbe nutrition, it's important to know that there are specific growth requirements that all microbes have. So first of all microbes are going to need a source of carbon. All of them are going to require a source of energy, and all of them need a source of electrons or hydrogen atoms. And we can classify microbes according to where they get these different sources. If we classify them according to the source of carbon, we get autotrophs and heterotrophs. Autotrophs are going to be microbes that are able to produce their own carbon sources. So in other words, they're able to use something like carbon dioxide, and that's inorganic, and they can convert that into something that's organic. So they can make their own sugars. Heterotrophs aren't able to do that, so they require another organism to produce that first for them. So when we're talking about heterotrophs, they must eat another organism or dissolve another organism, but they're getting their nutrients from another source. If we talk about the energy requirements, we have two different ways of getting energy. We have what are called phototrophs. Phototrophs are going to get their energy from a light source, and then chemotrophs are going to get their energy from a chemical source. And then as far as the source of electrons or hydrogen ions go, we have what are called lithotrophs, and we also have organotrophs. Microbes are also going to vary in their overall need for oxygen. What we'll find is that there are some organisms that need oxygen all of the time, and then there's others that cannot have any oxygen at all. Obligate aerobes are going to be the ones that require a constant supply of oxygen. The reason for this is that they are going to use the oxygen for their electron transport chains, which is going to be a major way of producing ATP for them. So they need that constant supply of oxygen. If we were to relate this to humans, we would be obligate aerobes. We need oxygen in order to survive. There are also organisms that are called obligate anaerobes. Obligate anaerobes are not able to survive at all in the presence of oxygen. So not all oxygen is exactly the same, and that's why there are going to be differences in whether organisms need the oxygen to grow or whether they can't have oxygen present at all. So oxygen gas, what we find in the atmosphere is O2. Okay, so that would be also called molecular oxygen, and so that is what we require, what we need. And then you also have oxygen found in a lot of organic molecules like fatty acids or carbohydrates, and that's different. We also have some toxic forms of oxygen. And the reason why these forms of oxygen are going to be toxic is because they are strong oxidizing agents. And oxidizing agents are going to be things that steal electrons from molecules. And that's what makes these toxic. They are going to be stealing electrons from molecules, and that is going to cause a lot of cellular damage. So these are the four toxic forms of oxygen. We have singlet oxygen, superoxide radical, there's a peroxide anion, and a hydroxyl radical. So all of these are toxic. All of them have the ability to steal electrons and cause cellular damage. But if we have organisms that survive in the presence of oxygen, the reason that they're able to do that is that they do contain special enzymes or pigments that help them to neutralize these toxic forms. So they basically take them and they're able to turn them into a non-toxic form. If an organism does not have the special enzymes and they don't have the pigments to deal with these, then they're not going to be able to survive in the presence of oxygen because they would just get too much damage and it would overall end up killing them. So if we were to look at how we can classify organisms, there are special tests using special media that help us to differentiate which microbes happen to be obligate aerobes or which ones are obligate anaerobes. If you look at these right here, the obligate aerobes would only grow in the presence of a lot of oxygen. So we see them right at the top of the tube here where there's lots of oxygen present. The obligate anaerobes, which cannot grow in the presence of oxygen, are going to be found way down at the bottom where there is not oxygen present. If we look at the other ones here, the facultative anaerobes, these are able to grow under both aerobic and anaerobic conditions. So they can grow both with oxygen and without oxygen. But for them, they do grow best in the presence of oxygen. For that reason, we will see an extra concentration of them up where you do have the higher oxygen levels. If we're talking about the aerotolerant anaerobes, these do not use aerobic metabolism, and they are able to tolerate the presence of the oxygen because they do have some of those detoxifying or neutralizing enzymes there. But since they don't really use the aerobic metabolism, even though they tolerate oxygen's presence, they're not really going to grow better in the presence of oxygen. So for that reason, we see a nice even concentration of their growth throughout this particular tube. We also have some microbes that require very low levels of oxygen. And for those, they're usually needing oxygen levels between 2 and 10%, and we call those microaerophiles. Other nutrient requirements are nitrogen. Nitrogen is really important for amino acids and then also nucleic acids. Amino acids are going to be used to build proteins, and we need proteins in microbes to help with transport. They're going to also be enzymes and run various reactions inside the cell. And then nucleic acids, that would be the genetic information, that would be the DNA. And then it also is going to be the RNA that is utilized when we do get ready to make some proteins. The nitrogen can be obtained from organic and then also inorganic sources. So if it's inorganic sources, that would be from nitrate and then also ammonium. We do have some special organisms that are able, in some cases, to actually collect atmospheric nitrogen. And this atmospheric nitrogen or nitrogen gas is N2. These organisms are called nitrogen-fixing organisms or nitrogen-fixing bacteria. And these are often used for crop growth. They're also, in many cases, found associated with plant roots, and they'll be very beneficial to plants because they do help them to get the nitrogen that they need because they are able to collect the nitrogen from the atmosphere, and the plant's not able to do that on its own. Other requirements that we do have, phosphorus is very much needed. This is going to be an important component of the membranes of the cells. It's also important for nucleic acids and for ATP synthesis. Some proteins also have some phosphorus in them. And then sulfur is needed for some amino acids and vitamins too. This is not an exhaustive list of the nutrient requirements. There are others. The others would be needed in smaller quantities, and we could obtain those in many cases from organic and inorganic sources. This is not an exhaustive list of the nutrients that would be required for growth, but these are going to be the ones that are needed in the greatest abundance. Other ones that are needed in lower quantities are called trace nutrients, and the cells could obtain those from a number of different sources. There are also going to be organic requirements, organic chemicals that are needed. These are going to be called growth factors. These, we have a number of them listed here. Growth factors, in some cases it's amino acids, sometimes it is actual steroids like cholesterol. You can also see that there are some vitamins listed on this list. You can see what these are needed for, and many of them are needed to make a variety of different molecules inside the cell, like nucleic acids and then also for proteins. Besides having specific chemical requirements for growth, microbes also have very specific physical requirements. At the top of the list for physical requirements is temperature and pH. This temperature and pH is going to have great impacts on the three-dimensional structure of biological molecules inside of cells. Right at the top, we can talk about proteins. Proteins have very specific three-dimensional shapes, and if we do alter the temperature or if we do alter the pH too much outside of the optimum range for that particular protein, it's going to cause the protein to unfold and lose its structure. And that would, of course, destroy the function of that particular protein. So this is the reason why we have strict temperature and pH requirements. This is going to be different for all the different microbes. So they're not going to have the same temperature requirements. If we look at one example here, here we can see that we have this temperature range for a particular microbe. It has an optimum temperature, which is where we see that highest peak, and then we also have a maximum. After that maximum temperature, the organism would not survive, and then we also have a minimum temperature where beyond that, below that, it's not going to survive either. We can see that growth is a lot less once we get outside of the optimum, and at the farther we get away from the optimum, we're going to have a lot less growth. Here we have another illustration of how the temperature is affecting the growth. For this particular one, the optimum growth is at 37 degrees Celsius. So if we look at that Petri plate, we can see the most bacterial growth on the plate. When we look at 30 degrees, it's not that much different, but it is less. But when we get over to 22 degrees, we can see that there is much, much less microbial growth in that case because we're really starting to get away from that optimal temperature range. Microbes can be grouped based on their preferred temperature ranges. So you can see the breakdown here of the temperature ranges. Now, the ones that we tend to find in our bodies, so both beneficial ones and then also disease-causing ones, they're going to be the mesophiles. So this is where most of the human pathogens are located at. And the reason for that is if you look at their ideal growth temperature, it's right at the same point that would be our body temperature. So they're going to survive well at our body temperature. If you look at the other three groups, they don't really have growth at our body temperature, and that's why you wouldn't really see them growing inside of our bodies or on the surface of our bodies. Here's a look at different groups of bacteria or groups of microbes classified according to ideal pH ranges. We have acidophiles, which need a very acidic pH. We have neutrophiles, which need more of a neutral pH, and then we have the alkaphiles, and they need more of a basic pH. And our bodies are primarily neutral or pretty close to neutral on the inside, which means that the neutrophiles, that's the group that's going to contain most human pathogens. Again, we're going to find human pathogens need the same general growth requirements that you find inside of the human body. Microbes are also going to need water, and their water requirements are based on two physical effects. First off, water is going to exert osmotic pressure. This is the pressure that's exerted on the membrane by the particular solution. So some solutions will cause a swelling, that's osmotic pressure, and so that swelling, how the microbe deals with that will help to determine what its water requirements are. And then we also have a hydrostatic pressure, which is the pressure exerted in proportion to the depth of the water. Some microbes will be able to survive and live at very deep depths. And again, this is going to be because they're able to deal with that really big hydrostatic pressure, which is all the water that is above them. Biofilms can live under extreme hydrostatic pressure. So these are going to be the ones that you do find at those very low water depths. If things are not able to withstand a high hydrostatic pressure, they would not be found very deep. Microbes can live in complex relationships with other organisms, and these relationships that they live in can be both negative or they can be positive relationships. In an antagonistic relationship, the microbe harms another organism. So that would be something like a pathogen. When we're talking about beneficial relationships, these are two types. We have what are called synergistic, and then we also have symbiotic. In the synergistic relationships, the individuals are cooperating with each other so that ultimately both of the participants, they do benefit from that relationship. In the symbiotic relationship, the organisms do live there with each other. They live interdependently with each other, but we're not seeing this mutual benefit from that relationship. Biofilms are a form of very complex relationships amongst numerous different microorganisms. This can be multiple prokaryotic organisms, it can be prokaryotic and eukaryotic organisms. But the key with this image here is just showing how they form. Basically, you have some initial microbes that settle on a surface and they start to secrete a sticky matrix, a glycocalyx over the top, which provides some protection for them. And over time, these microbes start to communicate with each other. This communication is chemical communication, It is what we call quorum sensing. So it's them sensing the presence of each other, and they can kind of sense their numbers. And once the numbers become enough, so they reach a certain level, the individual bacteria or the individual microbes start to differentiate themselves into different roles. So they begin to work together, and some of the microbes will perform a role that is more for like water channels. Some of them are going to probably be more for the production of carbohydrates or for collection of energy. So they all assume different roles for the benefit of this one big community that they are now a part of. We do see biofilms in some human problems. Dental plaque is one example of that, and that's what's being shown in this particular image here. When we're looking at plaque, plaque is a biofilm. It's a community of microbes that are working together in this symbiotic relationship.